Okay, here we go. We're going to cover the nucleus. Woohoo!
Let's not and say we did.
Okay, no, we really are. Okay, that's just the way it goes. It's fine, Moira.
I guess I'm stressed.
Okay. All right, the nucleus. Double membrane. What does that mean? 2 lipid bilayers. 2 phospholipid bilayers. Right? So if we were drawing... Look at me. No, not 3 legs. What is wrong with me? Glycerol, phosphate. Right? The little P guys. We're just going to do that. This is 1 bilayer. It is a double bilayer. I'm not drawing all the P's. Right? 2. There's 1 bilayer. There's another bilayer. The nuclear envelope. They just call it that. It's kind of stupid. It's just the nuclear membrane. Double membrane. Okay? This guy right there. Euchromatin. That's the stuff that's open that we do gene expression. Heterochromatin closed. No gene expression. The nucleolus. I don't know. That's where it's all packed in together. And then there's your stupid envelope, right? Double bilayer. Oh, well, interesting. All right, continuing, here we go.
Let's not and say we did.
Okay, stop it. Stop. Just stop. Okay, it's enough. Oh my God. Okay, knock it off. All right,
I guess I'm stressed.
Okay, we're all stressed. Stop it. Okay, double membrane, many nuclear pores. What is a pore? It's a hole, right? Pores in your face, your clogged pores. Oh, it's just a hole, right? So you can get stuff transported, move stuff in and out of the nucleus. Internal protein lattice, the nuclear lamina. So what do we think that's similar to? That would be like our cytoplasm. Cytoskeleton. Gosh, it's my problem. Maybe I'm stressed. Okay, so that's like the cytoskeleton in the nucleus, in the nuke, is the nuclear lamina. Okay, yes. Nucleus, multiple, yes, linear chromosomes. How many do we have? 46 chromosomes. 23 pairs of homologous chromosomes. We'll get to that later. Tightly packaged into chromatin, right? Only we call it chromatin when it's all in its regular state, which we would call spaghetti. And then it really compacts even more in order to see a chromosome. Only see these in mitosis and meiosis. We get to that. Woohoo! Okay, enough. Oh, yes, the nucleolus, I don't know how you say it, where the ribosomal subunits are assembled, large, ribo, small, ribo, right? Here's the little big and small. What does the ribosome do? Protein synthesis, translation. Makes proteins from what? Amino acids. Using what kind of bond? Peptide. Okay, so if we want to talk about moving in and out of the nucleus, we have that nuclear envelope, 2 membranes, right? So here we go, nuclear envelope meant bilayer 1, bilayer 2. It's actually continuous, woo, with the rough ER. We'll get to the endoplasmic reticulum in a second. Here's the cytoplasm, right? The liquid, it's a nice, like, little, I should have went, it's a liquid portion, right? Here's the inner membrane space of the nuclear envelope. And this is the inside. There's that lamina, like the cytoskeleton. This is the outside of the nucleus into the cytoplasm. Surface view, ooh, scanning electron microscopy, right? You can actually see these little pores, the little holes in the nuclear membrane, thousands of them. And a little cross section, same idea, scanning electron microscopy, and you can see it's a little hole. Here's that double layer inside. Right, here's the inside, here's one, here's the other. Here's some of the DNA. Interesting, stuck right there on that membrane. Here's the outside of the cell, cytoplasm. That's always outside. Outside nuke, yay. And, okay, we're gonna ignore the heterochromatin, whatever. Here's that inner membrane, right there, yeah. The envelope, double lipid, phospholipid. I'm going to say this 1,000 times so you know it's going to be on the exam. The bilayer. Bilayer. Inner membrane, outer is always towards the cytoplasm, inner towards the nucleus, right? Outside, inside. And then there's this pore complex, blah, 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 that lets stuff in and out. Let's look at it, shall we? Yes, we shall. Ooh! A macromolecular complex made of 30 to 50 proteins. It's a big, complicated mess. We're not going to memorize any of those proteins. Maybe in cell bio 380, who knows? Selectively permeable. Again, remember, what is the definition of that? Some stuff in and out, some not, right? It chooses. Well, the chemistry chooses, but whatever. Okay, so if a molecule, like a proteins, usually, imported, sent in, need a nuclear localization signal, NLS, what is that? It's just a sequence of amino acids in the protein. Somewhere has to have the specific sequence, I don't know what it is, some order, right, of amino acids specifically makes up an NLS. So any protein that wants to go in has to have this. If it doesn't have that specific sequence of amino acids, it's not going in. That's the rules. So it must be something recognizing this sequence, some molecule recognizes this sequence and lets it in. Well, let's take a look, shall we? Yes, we shall. Molecules shuttle to and from the nucleus through the nuclear pores, blah, blah, blah. Large proteins and RNAs must be specifically transported through the pores. Yay, we just said that. Same thing, right? If the proteins need to go in, they have to have an NLS. So this must have a specific sequence of amino acids in it to get sent in. We'll talk about coming out in a second. But as we know, I hope we remember, mRNAs are made, transcription happens in the nucleus, and then mRNA comes out, translation in the cytoplasm. So transcription as foreshadowing in nuke. Because it's DNA to RNA. The DNA is in the nucleus, so it has to be there. And then the translation is in the cytoplasm. And translation is RNA to protein. Woohoo! All right, we'll talk more about that later. So let's not worry about that right now. Okay, let's not stress out.
Sorry. I guess I'm stressed.
Okay, yeah, I know. It's going to be fine. We're going to go through it. Okay, So how do we get in? Ooh, they must have a zip code. Ooh, it's 17 amino acids. NLS. So the zip code is just that stupid sequence of amino acids specifically. I don't know what it is. I should probably look it up because that would maybe help us. But anyways, if it doesn't have 17 amino acids in that same exact order that makes up this, is just what we named it, then it can't get in. So they want to know, where is it? Where is this? Is it part of the main core, or is it part of the tail that allows it to get in? Like, where is this NLS is the question. Where is it? So where? So what do? So the specific protein that we know goes in, we know it goes in, and we want to know what part is it. Is it the core part of the protein, or are these little tails sticking off? What do you think? I would think it's a little tail sticking off because that's an easy way to add an NLS, but what do I know? So we attach a radioactive label. What does that mean? Well, we can see radioactivity as it breaks down. It gives off light and radiation, not so great, but you could detect light. Now we would tag it with a fluorescent dye that glows, which isn't dangerous for us. And then what they say, inject labeled protein fragments into the cytoplasm. Okay, so they put the tails in the cytoplasm, they put the core in the cytoplasm, and they want to see who goes in. And then we wait and see where they go, which goes through the pore. Woohoo! Okay, how are molecules imported into the nucleus? All right, so what is the prediction? Here's our hypothesis. Zip code is either in the tail or in the core. The null, the zip code is not in the protein, or there's none. Okay, what do we predict? What do you think it is? Do you think it's the tail or the core? And I don't care about the null hypothesis. I know I get in trouble for not caring about that, but let's just predict what we think is going to happen. Then you see this. What do you conclude? In class, upload, in class, homework, NLS, experiment. Pause the video and do this. Okay? Can you please do that? Okay, what do you think? Well, based on what we found, it looks like the tail fragments went in. And the core fragments are in the cytoplasm, not in. So my prediction was right. Tails have NLS. Woohoo! Conclusion, yes, tails have or are the NLS. Core does not have the NLS. So good.
Color me impressed.
Oh, thank you. Okay, so if we really want to know how, which we don't really want to know how, okay, I kind of do, because, I kind of dig this kind of stuff. Anyways, okay, so the protein that has the NLS has to work with another protein, the importin, and they named it that. They get together, and so the important probably sees the NLS, and when those two bind, then it can go through. Uses some GTP, and then, right, this complex goes back out so that we can use this important again. Okay, so that's what happens. GTP is hydrolyzed to GDP. Right, that means energy released. We broke off a phosphate. Woo! Whoops. So GTP is literally, right, guanidine triphosphate, ribonucleotide, can be used for energy like ATP. Ooh, so exciting. Okay, so if we really care, which this is from Cell Bio 380, So we're not going to, ooh, that's kind of nice. Bio 380 right there. But it shows us, because we only talked about the important, there's also an exportin to go out, right? And the mechanism similar, right? If it has a nuclear export signal, NES, again, series of amino acids, specifically to get out of the nucleus, you're going to get together with that guy. It's going to bring you out. You're going to need that GTP stuff, and then the guys are recycled. So same idea. You want to come in, right? Here's that nuclear import receptor that we call important, gets together with the guy with the NLS, comes in, right? There's our GTP. We deliver it, and then that recycles back out. So same thing. No details except important has to bind to the NLS guy. Exportin binds to the NES guy to go in. This is to go... Lies. Don't listen to me. I'm being an idiot. To go out. This one goes in. And the NLS and the NES, it's a sequence of amino acids. So this one obviously has to be different, right, than the NLS, because it's a different protein that binds, but that's what targets them. An NES to get out, an NLS to get in, important, exportant. Okay, so this other upload in the text box, tell me how we know what proteins get to go in or go out, right? I guess I answered it for you, but can you rewrite this? Please? Put it in the text box or write it out on a piece of paper, take a photo and upload. That would be spectacular, right? Remember, writing stuff down helps you remember. Looking at it, listening to me yap is not the same thing. So this is the end of our little tutorial on going in and out of the nucleus and the nucleus itself. So make sure you're taking notes from this stuff. Don't just listen to me, but also do it over and over, okay? And there you go. I'm out of here. So see ya.
I'm sorry. I guess I'm stressed.
Yeah, me too. Okay, we're good though. Bye. Oh, and that's it.